time, Professor Kaushik Chatterjee. To all before you, that Dr. Chatterjee received his PhD in bioengineering from the Pennsylvania State University, USA, after receiving MS in Material Science and Engineering from the University of Virginia, USA, and BE Metallurgy from Bengal Engineering College, Shivpur, India. He worked as a postdoctoral fellow jointly at the National Institute of Standards and Technology and the National Institute of Health, USA. He joined the Indian Institute of Science, Bangalore in 2011, where he is currently in a position of associate professor in the Department of Materials Engineering and associated with the Center for Biosystem Science and Engineering. His research group focuses on materials for biomedical applications. So with this CV, I request Professor Kaushik Chatterjee to start his presentation, as the time really is a very constant at this point of time. So I request you to be limited to only this half an hour, which is allotted for you. Yes, thank you, Professor Chatterjee, and you can start your presentation now.
Thank you. Thank you for the kind invitation, and it's a pleasure to interact with all of you, even if it's online. I've seen the work that comes out of CGCRI, and a lot of it overlaps with or complements what we do. So it's a privilege to be here. So what I wanted to do today was to talk about one aspect of our work, which involves polymeric nanofibers. And we've been looking at it both in terms of regeneration and also as platforms for disease models. So as a biomaterials lab, we've been interested in a variety of materials. We are, in fact, more sort of the materials of biomaterials, in the sense that we work on developing new materials and also looking into material processing issues. And so here in that context, we do a lot of work with metallic materials like titanium, stainless steel, and surface modification of these alloys to make them better for better materials or candidate materials for preparing orthopedic implants. I'm not going to talk about it today, but we also do a lot of work with tissue engineering or making scaffolds for tissue engineering. I don't think I need to introduce that to this audience, just to emphasize that you want to be able to take a biodegradable polymer, make it into some sort of a 3D structure in which you want to grow cells and form a tissue. And ideally, your material will not only support the tissue formation, but will also degrade as your tissue grows. So among the various ways of making scaffolds, one of the more popular methods in this field is this strategy of using electrospinning to make nanofibers out of biodegradable polymers. And I'm going to assume that most people here know. So just a very quick overview of this is that you take typically a polymer that will go into a volatile solvent, load it in a syringe, you apply a high electric voltage here, and then there is some physics involved in terms of forming a Taylor cone that you end up generating fine fibers of this polymer, and that's collected on a conducting substrate, which is your collector. As such, it will look like a sheet of paper or a very thin piece of cloth. But in fact, if you look at it in a microscope, you would find that these are mats which have these fibrous structures, which are typically in the size of hundreds of nanometers. The reason this is particularly exciting is that if you look at animal or human tissues, the extracellular matrix, after removing the cells in these tissues, would basically have this fibrous structure. And so here what we're trying to do is to prepare these scaffolds that have similar sort of an architecture. The other advantage, or the reason it becomes quite an effective substrate, is that one is it of course has this architecture of the roughness. The other is by making this sort of a porous structure, you'll end up reducing the modulus compared to, say, a block of this polymer, so that the modulus now is closer to that of human tissues. So we've been doing a lot of this work with polycaprolactone simply because it's quite conducive for processing. And also it is a biodegradable polymer that's been used in a lot of medical products. So as you can see here, it has many advantages, including the ease of processing, and also the fact that it's non-toxic, which means that you can grow cells. However, even though it's non-toxic, it doesn't have inherently any bone-inducing ability, which is where this processing and also modification of this polymer becomes important. If you look at the structure of bone, you would notice that, a lot of it is actually these mats of fibrous structures. And of course, these are, polymer fibers of collagen with a lot of ceramic in it, which is what is the structure of bone. And so the idea was to look at these scaffolds, first just the polymer fiber itself, and then incorporate ceramic nanoparticles in it to sort of mimic this ultrastructure of bone. This is some work of a former PhD student who did this work, where the first thing that we did was to start with PCL, make it into a flat film, and separately we made it into a nanofiber mat. We took human mesenchymal stem cells that were derived from the bone marrow. They were cultured on these scaffolds and also on the film. On the 2D film, of course, you get mostly a stem-like state of the cell. You do not get much osteogenesis. If you add osteogenic supplements to the media, you get high amount of osteogenesis. What we and others in the field have found is that even if you don't add the supplements, but you grow the cells on the nanofibers, there also you see increased expression of osteogenic markers, which is revealed by these images or these plots here. You can see here that the nanofiber is better than the flat film, even in the absence of osteogenic markers in terms of mineral quantification and also expression of several characteristic osteogenic markers or proteins. So we started to look into why that happens or what would be driving it. So the number one thing that we realize is that there is altered cytoskeletal arrangement when you put cells on these nanofibers. So you can see how the cells look very different when they are on the fat film versus they are on these nanofibrous mats. So basically the cells are narrower and also more compact. In fact, it's not just the cell itself, but also the nuclear morphology. And there is a redistribution or rearrangement of a lot of cytoskeleton, in particular actin and microtubules that we looked at in these studies. So particularly we were interested in looking at what could be the role of microtubules and if it's different between 2D and 3D. So when we stained for that, and also then we looked at a drug which is known to disrupt microtubules. What we found is if you add that drug at sublethal concentrations to the cells, what you find is that cells do change their shape, but it is less of a change or less profound if it's in 2D. However, in 3D, you get much larger change in the cell shape and size if you add this drug to the cells that are grown on the nanofibers. So the next thing that we did was to also look at what happens if you add in, what happens to the shape of the nucleus itself. And what we found is there is difference in terms of the shape of the nucleus. The nuclei are smaller and more elongated when you have the cells growing on the 3D compared to on the cells in 2D. And you can add in the drug to disrupt either actin or microtubules, and you find that there will be more of an effect on this. The microtubule disruption causes an effect on 3D, but not so much in 2D. Actin, of course, seems to play a role in affecting the nuclear shape both in 2D and 3D, right? So then the question was, if the shape and size of the nuclei is changing, what happens in terms of the heterochromatin packing or organization? So here what we've done is we stained for a protein called heterochromatin protein 1 or HP1. And by staining, what we found is that if you look at cells in 2D, which are in this so-called stem-like state, even, and that you would notice here that the antibody stains it in some sort of a diffuse manner, versus the cells in 3D, you can see these very bright, sharp peaks or spots, which are not so well distributed. So this is more sort of the differentiated state versus on 2D, it's a stem-like state. When you add the drugs to the cells in 3D, you sort of go back to the stem-like state, which then basically means that when you have cells growing in 3D, you alter the nuclear morphology, which in turn affects the heterochromatin packing, and that affects the gene expression. And so when you have cells in 2D, they are in the stem-like state. In 3D, they are in the differentiated state. And when you disrupt the microtubules, it seems to affect that, and they seems to revert back to the stem-like state, which is what we see in the case of differentiation by looking at the amount of mineralization or the amount of mineral deposits on the nanofibers. So the big picture really now is that why do the cells behave differently on 2D films versus on nanofibers? That's because on nanofibers, the cell shape and spreading is constrained. That affects the nuclear morphology, the organization of heterochromatin, and finally the cell differentiation. So the other aspect of this work was to see, well, now that we understand or agree that yes, the nanofibers are better than the 2D films, what could you do to further stimulate the bone regeneration ability? So of course, like many other people in this field, what we've been looking at is to add in various kinds of ceramic nanoparticles. And that's simply because it's more conducive for processing with electrospinning, unlike adding in biomolecules, which would be labile and more difficult to process in an organic solvent. So in particular, we looked into this strontium carbonate because strontium ions are known to stimulate bone cells. They can downregulate the activity of osteoclasts and upregulate the activity of osteoblasts. They've been used as anti-osteoporotic drugs. They've been added in biomaterials. They've been used as supplements for bone regeneration. So what we really did was to take nanoparticles of strontium carbonate. We prepared nanofibers of PCl alone or with 10 and 20% strontium carbonate. And when if you take these composites and you put it in an aqueous buffer, what you find is that there is release of strontium ions from these composites. It does seem to affect cell proliferation marginally, but not in a big manner. However, there's a bigger difference when it comes to differentiation. So here you're looking at calcium staining using alizarin red. So you can see that if you have 20% strontium carbonate, the mineral stain goes up significantly. And also that is further corroborated with all these other other osteogenic markers that are expressed more, suggesting then that if you have these ceramic nanoparticles, in particular particles that elute strontium ions, then they will stimulate the mesenchymal stem cells and induce not only just enhanced proliferation, but also increase osteogenesis. So there've been, we've done a few other studies. I won't get into all of them, but this one I wanted to showcase that there's, one important aspect of any of this tissue regeneration is the ability to stimulate angiogenesis or vascularization. So in that context, we looked at this ability to add in silica into these nanofibers. So we started, like in the previous study, by incorporating some silica nanoparticles in the polymer solution, and when we tried to electrospin, we realized that it was difficult to do that. The particles would agglomerate. So what we had to do was to get into a different kind of a strategy, which is in situ silication. But before I come to that, I just wanted to showcase here that how silica itself is a promising ceramic in that context, because it's inherently a minor constituent of many hard tissues, and it helps bone repair through angiogenic and osteogenic stimulation. So like I said, we had to come up with a modified or adopted this sol gel method in a manner that would be compatible with electrospinning. So basically, we took a source of silicon and we added some of these additives to adjust the pH. We tried with acetic acid or citric acid for comparison. And what we found is when we add these in, you can see here in these two vials, there is in situ precipitation of the silica particles that happens. And in a matter of 15 to 30 minutes, you get a good dispersion of silica particles in the solution that we can electrospin. So we characterized the particles and we found that these are some 200 nanometers or so, and these were amorphous in character. And also we were able to electrospin these, and you can see here that whether you use acetic acid or citric acid as the precursor or as the catalyst here, you can get the particles embedded here in the nanofibers. And if you put them in aqueous buffer, they will release silicon ions. Also, we did a lot of detailed characterization, which I'm sort of skipping over in the interest of time, but we confirmed that yes, you indeed get about 5 to 6% of silica in this system. And also, if you were to put these fiber mats in simulated body fluid, you get a significant amount of apatite deposition on these fiber mats. So when we grew mesenchymal stem cells on it, we found that the ones with the silica are more efficient in inducing osteogenesis. That was confirmed by either mineral deposition and also expression of several of these osteogenic proteins. Very interestingly, it was not just the mesenchymal stem cells that were stimulated, but also the endothelial cells. So if you grow these cells on these mats, what you find is the ones that contain the silicon or elute the silicon ions, you will find that they have a higher tendency to form these networks, which is a functional hallmark of angiogenesis. And you can confirm that with gene expression and protein expression studies. So basically what we were saying is that by incorporating these silica particles in situ in these electrospun nanofibers, you get several benefits. In particular, you can stimulate osteogenic activity and angiogenic activity together in the same in that same fiber mat. So we have tried this with other kinds of systems, which I will not talk about here. You're welcome to look up the papers. Here's something like bioglass, where we have calcium and silicon all together. This is an amorphous state, which in a similar strategy of doing in situ gelation, and that also gives these similar benefits. Okay. So aside from doing a lot of work on bone regeneration, we've also used these fiber mats, electrospun nanofiber mats in other applications. I want to showcase one study for cardiac regeneration, which we all appreciate is a major killer of modern life. And so if you look, if you were to take cardiomyocytes, and these are mouse cardiomyocytes that were cultured, you would find that you are able to grow the cells, but they will not be arranged in a manner that would sort of mimic how these cells would grow in mouse heart or in the human heart, right? So that's where this spinning of nanofibers becomes important. The idea was to see whether one can take stem cells, grow them on these electrospun nanofiber mats, and get them to differentiate and also align them and organize them into a tissue-like architecture. So what we did here was to take PCL film in the form of a flat substrate. We prepared PCL in the form of PCL nanofibers, which were unaligned. We blended it with gelatin also in this random or unaligned architecture. And these two were aligned by using a drum collector. And as a comparison for a different kind of 3D structure, we made porous structures that were of two different pore sizes. And we grew mesenchymal stem cells and added factors that would induce cardiomyogenesis. So again, I'm skipping over a lot of the experimental details and the results, but I just wanted to highlight the role of the nanofibers, and in particular, the benefit of using aligned nanofibers in this case. So if you look at the expression of certain cardiac markers like connexin 43, desmin, and also myosin heavy chain, what you would find here is that the cells that were grown on the PCL film express little to no markers of cardiomyogenesis. If you grow them on these porous fiber, the porous scaffolds at the bottom, again, you get a similar story. However, if you grow them on the nanofibers, you can see there is high expression of these markers, and it is particularly high when you have these aligned nanofibers, either of PCL or when it is blended with gelatin. And this you can confirm with gene expression. And in fact, we wanted to functionally validate that. So what we did was basically take out the cells from the scaffold after 7 days of growing them on these nanofibers. We co-cultured them with cardiomyocytes that were isolated from neonatal mice. And if you look at this video very carefully, hopefully you can see that the cells are beating, which is basically, again, a functional readout that, yes, they are showing the hallmarks of cardiomyogenic differentiation. And then we also tried to probe into what would be doing that. So again, the role of the cytoskeleton becomes important. So we added drugs that disrupt the actin, the microtubules, and also ROC1. And we find that all of these are important in promoting the cardiomyogenic differentiation of the stem cells on these scaffolds. And also there's epigenetic factors that are important. So there's sirtuin-6 and also a protein H3K9AC that binds to sirtuin-6 that were perturbed when you grow the cells on the nanofibers. And this is mediated by the ERK and the AKT pathway. So basically, the long story short is that if you grow cells on these nanofibers, there is altered arrangement of the cytoskeleton, which affects, of course, how the integrins bind on to the substrate. And that in turn, through the AKT-ERK pathway, affects sirtuin-6 and affects epigenetic pathways to promote cardiomyogenesis of the stem cells on these scaffolds. So is it okay if I take 5 more minutes?
Yeah,
Maybe
okay.
Yeah.
Just
Okay.
for 5 minutes.
All right.
Okay.
Yeah, maybe I'll try to finish before that. So one
Great.
last example is the use of these nanofibers to look at not regeneration, but to engineer models that mimic human tissues on a lab scale, which is basically this idea of preparing organotypic models. So if you look at cells inside the human body, they have cell-cell and cell matrix interactions that maintain their form and function. But when you put them on these 2D petri dish cultures, these interactions are largely lost. And this is sort of recapitulated in an animal model, but they of course have several limitations. So the idea is to be able to make engineered substrates where you can recapitulate at least some of these interactions. So here we are looking particularly at breast cancer, which is one of the most common forms of cancer in women. And we were particularly looking at the role of stromal cells. These are cancer-associated fibroblasts in breast tumors. And they have several roles starting from ECM synthesis, stemness, inflammation, and so on. And what is well known is if you take these cancer-associated fibroblasts, grow them on a petri dish, they lose their tumor promoting activity. So the idea was, can you grow them on a substrate that you will maintain that tumorigenic activity? So our collaborators lab had been getting these breast tumors from patients who were getting the surgeries done from a hospital in Bangalore. And they had a protocol for specifically isolating out the fibroblasts from these tissues. And so what we did here was to prepare, again, these electrospun nanofiber mats, and we grew these cells on these fibers and compared them with cells that are grown in the 2D TC plates. And what you will notice here is that cells will locally align, but not globally if you have the cells on 2D, but on the PCL nanofiber mats, the cells will globally align. This is an important observation because what's known from a clinical standpoint is that cancer-associated fibroblasts will form these aligned arrangements, and this aids in invasion or makes it a more of an aggressive tumor if this kind of histological evidence is seen in those kind of tumors. So this plays an important role in sort of having a more aggressive tumor phenotype in these patients. So then we checked for other details, basically that if you have the cells in 2D versus on the fiber mats, you see more expression of alpha smooth muscle actin and fibronectin, which is an ECM protein. And also if you take the media that is of calves that are grown in 2D or from 3D, and you put it on MCF7, which are breast epithelial cells, you find that the cells, these breast cancer cells, will grow more or faster when you get the media from 3D, right? Which means that the calves, when they're on the nanofibers, have a more aggressive phenotype. And there's also been believed that the calves are involved in inflammation that is mediated by the NF-kappa B pathway. So what we found here is when you grow the cells on these nanofiber mats, they have higher expression of P65, which is a subunit of this NF-kappa B complex, which suggests then that they are showing increased expression of or hallmarks of cancer inflammation. And also downstream of that NF-kappa B, several other cytokines have been perturbed. So the last evidence of this sort of aggressive phenotype is what we did here, is that we took the calves which were either grown in 2D or 3D, mixed them with breast tumor cells, and co-injected them in breast tissues of immunocompromised mice. And after 45 days, the mice were sacrificed, and what we did was to take out these breast tumors that had formed there. And what you will notice here is the size is not very different. There was no statistical difference. However, what was observed is that when you have the calves from 2D, it forms a less aggressive tumor. So it is localized within the mammary fat pad where they were injected. However, if you have the cells from 3D, they go out of the mammary fat pad into the muscle, the subdermal fat, and you can see that the tumor has invaded all the way out to the edges of the skin. So, and when we showed these slides that to pathologists who were blinded to the study, they observed more inflammation in these tumors when they were from 3D compared to the ones in 2D. So essentially what we are saying is that the nanofiber mat could be a better platform for maintaining the tumorigenic activity of cancer-associated fibroblasts and could be a better way for drug screening for breast tumor or other kinds of cancer. So let me end there by thanking a lot of people who've contributed to the work over the years, from former students to current students, and also a lot of collaborators across campus. In particular, the work with looking at the cytoskeleton of mesenchymal stem cells was done with Vaishnavi. Ravi is helped out, or he's our collaborator on the cardio work, and Anu's lab does all the cancer work with whom we collaborate on the breast tumor models. And of course, all the funding agencies who support us, and also all the various students who've contributed to this over the years. So thank you again, and I'll be happy to answer questions now or later. My email address is written here. Thank you.
Yeah. Thank you, Dr. Chatterjee. Yeah, as we see that we have one question in the chat boxes just there, and it was a really very fantastic presentation. The more we see your slide, the more our curiosity. So what is exactly mentioned in the next slide? The amount of science, then the level of understanding, the scientific part in your presentations, and so really it's a praiseworthy, and just one cannot believe it. Yeah, so much amount of science and really scientific attempt and scientific tools and techniques you have implemented in your research work, and that's the really it has been a good work. We are really very thankful for your very illustrative presentation, and really it has been a lot, and definitely the students and as a whole the total community who are here may be enriching with the knowledge base with your esteemed presentation. Thank you so much for your kind consent and give a time with us at this point of time. We started the day with Professor Bikramjit Basu with the deliberations from Indian Institute of Science, Bangalore. We ended this particular session with the ending of the talk, very fantastic talk from Dr. Kaushik Chatterjee of the same organization, Indian Institute of Science, Bangalore. So I think Dr. Jui Chakraborty is, she wants to say something, or she is having some query. Dr. Jui Chakraborty, please.
Many others are having, sir. Actually, Dr. Chatterjee, a small query, like because there is a very, very big time constraint. So small query is like you are adding silica particles for angiogenesis, but have you thought about its toxicity?
Yes, so we are, yeah, I mean, those are valid concerns. And of course, I cannot claim that we have done extensive, toxicity studies. So at least the in vitro studies we have done, we did not find any problems because we are working with, in that particular study, we are working with primary mesenchymal stem cells and also primary endothelial cells, which are generally quite sensitive to toxicity. So there were, at least in those limited studies we have done, we have not observed toxicities. For those particular scaffolds, we have not done an animal study. So that will have to be done before it can be used clinically.
Okay, Professor Chatterjee, thank you. And please, all the viewers, we want to take a snapshot.
Yeah.
Any more question?
Yeah, this is Shubhadeep.
Yeah, Shubhadeep is here. Shubhadeep, yes.
Yeah, real quick. I have one question just here. Kaushik, thanks a lot for your nice talk, and also thanks a lot for accepting our request, our invitation to deliver this presentation. Just one quick question. Have you thought of checking the cell organelle on the 3D nanofiber cells? Do you think it has any influence on the potency that why you're forming osteogenesis, why the 2D surface is not forming? So
right.
The shape and size of the organ.
Right. No, so I think, I'm sure it is changing because if the cytoskeleton changes that much, I'm sure a lot of the other organelles would also have changed. it's certainly worth looking into. We have not, and in all honesty, there are so many questions that, and it all depends on which way this, the students take their research work. So that maybe one day I'll find another student who would be curious to do that, and we'll take it up. But right now we have not looked into it.
Thank you.
Okay, so we will take a snap all together, like all the viewers, please make your videos on. Yeah, others? One more. Okay, thank you all. Thank you, Professor Chatterjee.
Yeah, thank you for the invitation.
Yeah.
Thank you for excellent talk, Professor Chatterjee.
So thank you.